All right, happy Thanksgiving week to everyone. This is fall 2020. We are in week 14. Today is our only live lecture for the week. We do have some online homework, ADILT labs to get done. That'll count for our attendance for lab on Tuesday and for those online attendance for next Monday. Okay, attendance for today was taken by, again, your ADILT labs that you had submitted that were due last night. Those grades have been updated. All right, so we are on acid and base, the last part of the chapter 27 material, and I'm going to switch to the whiteboard that will let me write a little bit easier on all of this material. Okay, so we did electrolyte, we did water. Hopefully everybody got a really good review of some of the reasons why we care about electrolytes, we care about water balance. All right, and so now we need to talk about acid and base balance. All right, now this first little part here is a review of what is an acid. Okay, an acid is a molecule that when you put in water, so this is water background here, all right? An acid is a molecule that when you put it in water, it will donate a proton, meaning, all right, it is going to give you a free hydrogen, and what's left is going to be some type of molecule with a negative charge, okay? And so our body makes a ton of acids. Okay, we actually make a ton of weak acids, and then we do send into our stomach. Remember, our stomach is full of hydrochloric acid, which means that in our stomach, we are going to send a hydrogen molecule and a chloride molecule into the lumen. And again, technically, they will in random motion combine to form hydrochloric acid, but because most of the stomach is, again, a watery solution, most of the hydrogens and the chlorides will be freely separated independent of each other, and so that is why it's considered a strong acid. Okay, we make actually a lot of weak acids, and again, things like amino acids, again, many of our proteins, because they're made up of amino acids, are weak acids. We make free fatty acids. We have lactic acid. So we make a lot of weak acids, which means that when we put those molecules in water, and again, there's water inside our cell, there's water outside our cell, many of those molecules, again, in constant motion, are going to sometimes see liberation of a hydrogen, and what's left will be a negatively charged molecule that is suspended in the interstitial fluid, extracellular fluid, the plasma body water, or inside our cells in the intracellular water. Okay, so because many of the molecules that we have in our cells, outside our cells, traveling in our blood are some type of acid, okay, we then have to make sure that all of these free hydrogens do not accumulate to make our pH scale. Remember, our pH scale is the number of free hydrogens, and 7 is considered neutral, right? And we like to live right here at 7.4 in our blood water and in our extracellular space. So we like to be slightly slightly basic, which means we don't like a lot of free hydrogens floating in the waters of our body. Okay? Now, we are going to make acid show up in our body, usually from 3 different types of pools, okay? Some of the acids that show up in our body are known as fixed acids, okay? That means that once they are produced, they remain in our body fluids until they are eliminated by the kidneys. Some of these are some type of sulfuric acid and phosphoric acid. And again, those come from, many cases, proteins, amino acids, and phospholipids, all right? And again, there's a protein that has that amine group, and again, that carboxyl group. It's R group. One of them has these SH, and again, the SH, when you put it in water, tends to donate that hydrogen. And so anytime I take this certain amino acid and I break it apart and do something, I liberate potentially some of these sulfuric acids, or again, when I do with phosphate, and I liberate some of the phosphates from DNA, from, again, my phospholipids. So remember, there's glycerol has 3 carbons. All right, 2 of those carbons will be bound to a free fatty acid. One of them is bound to this phosphate molecule, and that's why it's a phospholipid. When I break this up and I get glycerol, I get the free fatty acids, and I use those to make energy, all right, I get left with this phosphate. And so that's how I can end up with some phosphoric acid. And then nucleic acids, again, they'll have amine groups in the A, the G, the T, the C structures. And remember the background of how these molecules, you know, you draw them like this, A, C, T, G, and then the other one you draw T, G, A, G. These little lines represent phosphates that are binding these nucleic acids to each other. And so anytime I start to break apart DNA and RNA, I'm going to have extra phosphates that are in my system. Okay, so that's a lot of how I end up with potentially acids in my body that I kind of like have to get rid of them, because they get to a place where I can't use them anymore because I've broken them off or I've torn them away from useful models, and now I have these kind of pieces and parts of those formerly useful models that I need to now get rid of. And while those molecules are suspended in my plasma, in my intracellular or extracellular fluid, they are an acid in that they are donating a hydrogen to the free hydrogen population as they are suspended in that body water. Okay? Now, organic acids are going to be, again, when I break down glucose, and glucose is 6 carbons, all right, I cut it in half, and that gives me 2 molecules of pyruvate. And technically, pyruvate, we sometimes hear it called pyruvic acid, right? And then pyruvate, again, when oxygen is not easily available, pyruvate, remember, can be formed into lactate or lactic acid. And so those are molecules that temporarily they are suspended in my body water, donating a hydrogen, but they can be used and broken down further. It's just they are in this suspended intermediate state where I could potentially build up the number of them because of bottlenecking in the mitochondria and the timing it takes and the number of, let's say, checkout counters I have to continue to break them down, or overloaded and I'm beginning to get back up in the line and accumulation of these molecules. Okay, normally if I give time, all right, and oxygen continues to come in, and this is what with exercise we cause that post-exercise oxygen debt, all right? If I continue to, again, take in the oxygen, continue to run the ramp up the system, I can potentially remove and finish completing the breakdown process of the molecules, or send them to the liver and reconstitute them back into glucose through gluconeogenesis, thus removing them from the acid load that I'm carrying. All right, so these are the acids that if I take a marathon runner, or I take someone who's just completed some type of very physically demanding event, so let's say a cross-country runner, and they ran hard for 5-10 miles, and they ran where they were pushing their body to the limit, and they were skirting that area of aerobic conditioning, anaerobic threshold. Chances are if I take blood from that athlete and I pull it the second they finish the race, chances are they are probably slightly acidotic, all right? They're acid rich because they have, for that trying to finish the race and trying to push the body as high intensity as possible, they have probably accumulated lots of lactate and pyruvate, and it's caused their acidity of their blood, the acidity of their extracellular fluid, and the acidity of their muscle cells to probably slightly drop from the ideal 7.4 to maybe even 7 and even lower. But they're not going to turn around and die on me right there, or normally they won't die on me right there if they're acidotic. It's just you give them a few minutes and things will slowly start to recalibrate back to normal, and they'll start to, again, feel better after they catch their breath and they let things reset. Okay, so those are a different again, pool of acids that we make and we accumulate, especially when the availability of oxygen becomes limited because of how much we can deliver and how many mitochondrial lanes we have and how many, again, fuels we can burn in a given second. All right, the last pool is that carbon dioxide. So it's tied to, on the end of the pyruvate and the lactate and the glucose breakdown and the end of fatty acid breakdown, when I've liberated all these carbon dioxide molecules, I now need to try to transport as many carbon dioxide molecules as possible to my lungs and blow them out. Well, to get them from the tissues and the cells and the cytoplasm of my cells into the plasma and then into the lungs, I need to, again, convert carbon dioxide to another molecule, because carbon dioxide is limited in how much it can be saturated in the plasma by Henry's law and how much can bind to open seats available in hemoglobin, because oxygen has vacated some of those binding spots, but not every binding spot. So I need to get a crap load of carbon dioxide converted into, again, with water, converted into hydrogen ions and bicarb. All right, and again, I don't want my free hydrogens to add too much to my pool, so I usually try to bind these up to keep my free hydrogens low, so I stay mostly alkaline. And these guys, remember, I kick into the plasma water, so that way any other hydrogens that accumulate in the plasma water from my organic acids or my fixed acids, I can try to bind up some of those hydrogens that are free floating, convert back to carbon dioxide, and again try to maintain the low levels of free hydrogens and the pH slightly alkaline. Okay? So anytime one of these pools of acids becomes very, very high than normal, I run a risk of possibly my body becoming more acidotic. All right, so when you talk about acidosis, a condition where you have a pH that's less than 7.35, and it's because one of these pools of acid populations increases the free hydrogen population in your body water, that can lead to a sense of acidosis. All right, now, because organic acids and fixed acids are related to potentially making too many of these or removal by the liver and kidneys, these types of acid accumulations we associate when they are causing the acidosis as metabolic in nature. And then anytime the carbonic acid population increases because the respiratory rate is not sufficient to get rid of or keep enough carbon dioxide, usually that is going to be a respiratory cause of acidosis. All right. Now, because I'm making acid and I have a crap load of acid and I want to keep my hydrogens very much bound up, I want to keep bound up hydrogens in my water of my cells, in the plasma of my extracellular fluid, and in my plasma of my blood, I need molecules that can bind them. All right, and molecules that bind excess hydrogens are known as buffers. Okay, and I have three big buffer populations in my system. Okay, the three buffer systems, three buffering molecules that I utilize are phosphate molecules, because again, phosphate molecules tend to have this negative, so they could potentially bind to a positive and together make a neutral molecule. Okay, proteins. Again, many of my proteins have these carboxyl groups, they have amine groups, and many of these carboxyl groups exist in solution of my body waters already negatively charged. So if I can make them bind up and convert back to this neutral molecule, I can then take some of these free hydrogens and keep them from being very highly populated and free floating in my body water. Amines could actually, again, take a extra hydrogen on, bind it up, become a positive molecule, but this type of positive molecule is not going to make pH stay acidic. So it's a buffer that can bind up some free hydrogens, become a positively charged molecule, but not affect my pH levels. Okay, so that because these are molecules that are heavily available in proteins because they're part of amino acids, proteins give me a lot of buffering ability. Okay, and I have proteins, of course, in my cells. I have proteins on my membranes of organelles in my cells, proteins free floating as enzymes in my cells, proteins inside my cells as structures like actin and myosin. So I have a lot of structural proteins embedded inside my cell. I have proteins outside my cell, some of them being free enzymatic molecules, hormones, some of them being carrier proteins. I don't have maybe as many outside my cell, but again, there are some there. Okay, and for hemoglobin, remember, hemoglobin is a protein that I have in high amounts inside my red blood cells. So hemoglobin inside my red blood cell is an important protein buffer that is going to not only keep the pH of the red blood cell at 7.4, but it's going to help also because hemoglobin can bind free hydrogens that are created through the carbonic acid being created of carbon dioxide and water binding and forming hydrogen and bicarb, that I can also influence by hemoglobin binding up hydrogens. I can kick out the bicarb and help the plasma levels be buffered by binding up hydrogens that end up in my red blood cells. Okay, so hemoglobin is an important buffer that is both inside cells and can influence by the kick out of the bicarb, all right, the the HCO3 negative can also influence extracellular fluids. Okay. And then my last buffering system is those HCO3 negative molecules coming from carbon dioxide and water breaking up into, again, HCO3 negative. These bicarb molecules, because remember, some of these free hydrogens are going to be bound up by my proteins and my free floating amino acids, and that's going to leave excess bicarb. So as I unload maybe other acids from other sources and have more hydrogens that show up from other locations, I can try to bind them up and again in the lungs get that CO2 out, therefore getting more and more hydrogens that would be free from these other sources tied up as water molecules. Okay, so these are my three buffering systems. And again, to go through them in a little bit more detail, let's start with those proteins. Okay, and again, this picture here, let me kind of clean it up around it, all right, shows you that normally, all right, this is what proteins for a central carbon, all right, is bound to a carboxyl group. And again, that hydrogen can be there. And if you put it in water, because this is a weak acid, all right, when you put it in water, sometimes that carboxyl group, that hydrogen disassociates, all right? And that is part of the reason why we call amino acids an acid, is because this carboxyl group can sometimes have the hydrogen there and be neutral, and other times in water, depending upon the hydrogen ion solution amounts, could potentially be free of that hydrogen and negatively charged. Okay. The other thing to remember is this R group, it may have more carboxyl groups, it may have more amine groups, it may have those sulfur groups, it may have some other chemistry related molecules you talk about in chemistry class that again at times could be donating hydrogens or gaining hydrogens, all right, and therefore playing a role when the free hydrogen levels start to accumulate, helping us potentially bind up, soak up, and absorb some of these hydrogens, or in other situations, giving us hydrogens when we need them to make pH stay steady. Okay, so normally when, again, let's say I've made a lot of lactate and lactate has become a hydrogen donor. I'm also creating lots of carbon dioxide because I'm burning fuel as fast as I can. So carbon dioxide and water are giving me lots of hydrogens and bicarb, all right? So what happens is inside my cells, as these free hydrogens accumulate because of the lactate, because of the pyruvate, because of the carbon dioxide, all right, that hydrogen population, as it rises, I am going to take whatever hydrogens I can and put them wherever there's an opportunity to place them. So if I have some carboxyls that are negative, I'm going to bind that hydrogen back to it. If I have, again, some hydrogens that I can even bind up and put in these NH3s, I'm going to do that. Okay, so I am going to try and put these free hydrogens I'm creating as I make a lot of lactate, as I make a lot of carbon dioxide, as I make a lot of fixed organic and volatile carbonic acids, I am going to try to put them in every place that I possibly can that a protein has available to try and bind up and neutralize or even make protein slightly positive. And in doing so, I keep my free, all right, hydrogen numbers, so the pH stays at about 7.4. Okay, when, let's say, I am hyperventilating and I'm breathing out carbon dioxide as fast as I make it, and I am potentially seeing that more and more and more and more of my free hydrogens are combining with bicarb and becoming water molecules as carbon dioxide leaves. If the pH ever gets too alkaline, again, any of these potential places in amino acids and proteins that are holding hydrogens in these spots where that I could take them away and add to my, again, free hydrogen population to keep my pH around happy 7.4, I can then do that as well. So proteins, amino acids, many molecules that have these, again, carboxyl groups, these amine groups, they are going to be very helpful in situations where the pH starts to become more acidic as I create more acid with activity, with breakdown of DNA, DNA, RNA, phospholipids, breakdown of cells, breakdown more glucose, breakdown and bottleneck my mitochondria with pyruvate and lactic acid. I can then absorb those excess hydrogens in lots of little places, lots of little nooks and crannies, and create a free hydrogen environment that stays at 7.4. On the flip side, anytime the hydrogens become, again, such that I need to get hydrogens away from all these nooks and crannies and put them in my free population so my pH doesn't become too basic. Again, many of these amino acids and proteins are weak acids and they're holding hydrogens bound up in a neutral molecule form, and I can pull those hydrogens off as I need and create, again, my pH to stay as stable as possible near that 7.4. Okay, so that's how the amino acids, proteins work. And remember, amino acids and proteins are located in cells and out of cells. All right, let's go to the phosphate one next. All right, the phosphate buffer system, it's going to work very similar. All right, the difference is I usually only have phosphates inside cells because I use phosphates again to make adenine into AMP and then add another phosphate, ADP, and then I add another phosphate and I make ATP. Same thing with G. I make guanine into GMP, GDP, and then GTP. Okay, so I need phosphates to be able to make all these nucleic acids into these different molecules. I need phosphates again to bind to glycerol with 2 fatty acids to create phospholipids. And anytime I want to make more membranes, again, whether that's for vesicles or for new organelles, all right, I need phospholipids. So phosphates tend to be, again, high inside the cell. Now, phosphates are only going to help me when the cell is making a lot of acid inside the cell. Phosphate buffers are only going to help, again, one cell and just the inside of the cell. So if this one cell starts creating a lot of acid from the breakdown, again, of glucose, and then its little mitochondria are backed up, and its lanes of turning pyruvate into carbon dioxide and ATP are bottlenecked, you know, like checkout counters on Black Friday or some shopping day, all right, and the acid levels start to accumulate, the phosphate is only a temporary help, okay? It's only going to help me bind up the hydrogens for as long as this cell has available phosphate to bind up the hydrogens that this cell is making and creating as it's dealing with its metabolic issues, okay? So this buffer system is only good inside one cell at a time. It has no effect on the hydrogen ions outside the cell. Okay, but think about, I'm doing my marathon. My skeletal muscles are potentially backlogged, can't get enough oxygen to keep the glucose burn going. They're having to make a lot of pyruvate into lactate. I'm still asking them to run at a super fast mile a pace, and those muscle cells are becoming slightly acidic. All right, the phosphate in there is going to keep the muscle cell from becoming acidotic. And then when I finish the race and hopefully breathe in extra oxygen and continue to run things for a few minutes to hours after the race and recover the debt of not having oxygen at times when I needed it, I can then, with these phosphates, temporarily keep the cell from being destroyed, from exploding, from becoming sick, while I finish the race, get the oxygen I need, finish breaking everything down and reconverting everything back into normal non-acidic donating molecules. Okay, so again, when it says it's temporary, it's only going to help that one cell for a small period of time. It doesn't eliminate any of these hydrogen ions, so eventually these hydrogen ions will have to be taken off of the hydrogen, reconstituted into the molecules maybe they came from, and those molecules like lactate might need to be converted back to pyruvate or converted back into glucose so they can be utilized later to make energy. Okay, now the other buffer system is, again, the carbonic acid, and the hemoglobin buffering system comes into play here, okay? And so again, remember that carbonic acid is that carbon dioxide is, again, easily able to diffuse across membranes of cells, go inside cells. The cells have water as their cytoplasm. So in the presence of a protein, which is carbonic anhydrase, carbonic acid is formed, and then hydrogens and bicarb are created because carbonic acid is like our little graphic earlier. When you put it in water, these two molecules separate. Okay, and the protons can be, again, any of the carboxyl groups. I could potentially bind the proton to those carboxyl groups in carbonic anhydrase. Any potential, again, amine group, I could bind and form NH3 positive and make my enzyme help me carry some of those hydrogens. All right, but because inside red blood cells, I have a ton of hemoglobin. Remember, hemoglobin is maybe binding oxygen, but it might only have 3 spots, so 4 is maybe binding up some of the carbon dioxide. All of these little areas might have some carboxyl groups, might have some, again, amine groups that these extra hydrogens being created can now be bound to, all right, and form neutral molecules or positive molecules. And to keep the hydrogens, again, bound up and not reforming with bicarb, I kick my bicarb out. And again, any lactate my cells kick out, and these hydrogens could potentially bind to that bicarb. And so hemoglobin buffering is letting me not only buffer the hydrogens from, again, carbon dioxide coming in and creating, but it's also helping me somewhat buffer some of the plasma by kicking out the bicarb and making the bicarb part of my extracellular fluid and water environment that is my plasma. So the hemoglobin buffer system, as it says, it is going to help inside cells, and it's inside the red blood cells, all right? But it's also going to help plasma, again, stay at 7.4. So this is the one protein system that has an effect immediately inside the cell it's in and on the outside extracellular fluid around it. Okay, so that is an important system that lets me not only help my red blood cells not die of acidosis, but it also keeps the blood that the red blood cells are living in, the plasma, also not get too acidic. And that is so critical for me as my red blood cells in my blood move, and then when I get into the kidneys, I pull that plasma out and I can dump those hydrogens and retain the bicarb, so the bicarb can continue to, again, buffer the extracellular fluid. And when I get to the lungs, that blood, again, can lead to the carbon dioxide coming out and hydrogens getting combined to form water molecules, and the carbon dioxide is gone, and I'm tying up more and more what would be free hydrogens into water molecules. Okay? All right, so this bicarb, I can't let too much bicarb exist in my system. So I do try to keep some of my bicarb in a stored form when and if I start to over accumulate it. All right, and remember, bicarb is a negative molecule tending to be, all right, and bicarb as a negative molecule can bind to positive things if needed. And so when and if I start to maybe have too much of this bicarb in and around my system, I will use the positive molecules I have in high amounts, which just happen to be in plasma, things like sodium, and I can bind it up, and again, in a bound form, that keeps the free bicarb available to keep my pH of my plasma at 7.4. All right, and so that is an important concept also of why I like bicarb as a buffer in my plasma, because I can, when I need to, also bind it up, temporarily reserve it for later when maybe the hydrogen ion population starts to accumulate again, as maybe I get going on another interval or get going on another workout, and I can then utilize that now free, which was previously bound to sodium bicarb, to help continue to buffer the system. Okay? All right, so let's talk about, again, when and if the hydrogen ions become higher or lower than normal, what happens? All right, so again, our goal is 7.4. All right, 7.45 and 7.35 are the range. Anytime the hydrogen ions decrease or the bicarbonate ions increase so much that my pH becomes greater than 7.4, we are going to call that alkalosis. Okay? On the flip side, anytime, again, the hydrogen ions increase or the bicarbonate ions decrease so much that the acidity drops below 7.34, we are in a state of acidosis. Okay, so this is where we want to be. these are the conditions when we go out of that range. All right. And remember, we usually use a few places. So the kidneys are one place that I can dump either hydrogen or bicarb. Okay. The stomach, remember, is a place where I put hydrogens, and the small intestines is a place where I put bicarb, again, to buffer chyme that comes in from the stomach. All right. So any kind of situation where the kidneys stop working and I don't get rid of the hydrogens or the bicarb effectively, that could be a reason why I end up with alkalosis or acidosis. Anytime potentially the stomach starts overproducing and I start making so much stomach acid that I decrease, again, my hydrogens free in the system, that can cause potentially alkalosis. And anytime I maybe have so much loss of contents from the small, which then is the large, and then through diarrhea, I could potentially get myself in a situation of I'm losing bicarb and I'm retaining hydrogens, and I don't have the bicarb to buffer them, so I can end up in a situation of acidosis. Okay. Normally, when these guys are the problem, we call it some type of metabolic condition. Okay. Now, when the situation is related to carbon dioxide being breathed out or breathed in, that is normally going to give us a situation of a respiratory induced condition. Okay. Now, right now, sitting at your desk, I want everyone to start pushing air out fast. All right, so that is called when you push air out fast. So I want you to breathe fast. That is called hyperventilation. And in hyperventilation, the CO2 is going to leave faster than you make it. Okay, so you are going to see that hyperventilation is one of the ways we can cause respiratory alkalosis, because we are going to breathe out CO2, tie up hydrogens into water molecules faster than we're making CO2, and CO2 is binding with water and forming hydrogen ions, and we are getting into a period of alkalosis. Okay, and so what can make us hyperventilate? Remember, stress, anxiety, or just being stupid and making ourselves do it on purpose. Right? You can potentially get hyperventilation when you are tachycardic, when you're high heart rate. You can get hyperventilation with high stress, hypoxia, when oxygen is not being available in the atmosphere and you are then breathing at a higher rate, trying to get more oxygen unloaded at a faster breathing rate with the blood moving in the system. All right. And again, when we become respiratory alkalosis, all right, if it continues, we will pass out. And our central nervous system is going to try to make us pass out to try to let us not breathe and build up carbon dioxide molecules so our pH can reset. So this is why people who hyperventilate could pass out, and they might actually have a pulse. They might actually have their cardiovascular system working, but they might not be breathing for a little while because they have expelled out so much carbon dioxide. The brain thinks that the system needs to reset and you need to stop breathing and build up carbon dioxide. Okay, this can happen, again, people like swimmers who don't want to breathe when they do their 50-meter race. They can hyperventilate to try to use that advantage of, if I don't breathe, I don't have to turn to the side and I don't lose my streamline. And then that can lead to them passing out in the water. And then when they wake up, their first thing they're going to do is try to take a breath. And if you take a breath underwater, you're going to take in water and not breathe. So it can lead to shallow water drowning. Scuba divers and free divers sometimes also hyperventilate because they think, I won't have the urge to breathe and I won't breathe my tank as fast, or I can go deeper if I don't have the urge to breathe and get into deeper depths. The problem is, if you then pass out, underwater and you're heavy and you fall, you're not getting closer to air, you're getting further away. So it could lead to drowning, or it could lead to, again, problems with, if you're scuba diving, again, problems with your tank, and you pass out and maybe your mouthpiece comes out of your mouth, and when you wake up, you are panicked and you are not able to get breath and find your mouthpiece. Okay. On the flip side, the opposite of hyperventilation is hypoventilation. And again, hypoventilation, there are certain drugs that can induce this. There are, again, trying to slow down your breath. You can make yourself try to do this. And then there are other causes with headache, with high potassium levels, because again, your membrane potentials are messed up, your action potential generation is messed up, and your brain is not able to tell the muscles of your diaphragm and intercostals to contract at a rate that is sufficient. And so again, it can cause very shallow, slow, and hypoxia to occur, and then now you're getting into killing off cells and dying cells because they're not getting oxygen and able to make their ATP. All right, so these are the sources, again, usually related to acidosis or alkalosis, the free hydrogen levels happening because we are breathing off the CO2 faster or not breathing off the CO2 fast enough. Okay, and if we are on a ventilator, or if we are in some type of lung disease state where that's the best we get is we're always hyperventilating or we're always slightly hypoventilating, then you will see that the kidneys will then try to compensate. All right, and the kidneys will compensate. If I'm hyperventilating, the kidneys are going to do a better job then of trying to retain acid for me and dump bicarb. And by retaining acid and dumping bicarb, the kidneys are going to try to correct for the air leaving faster with carbon dioxide and the ability of carbon dioxide to create acid for us. There, it's going to try to compensate for that. On the flip side, if I'm chronically hypoventilating, I am going to try to make the kidneys dump more hydrogen ions and retain more bicarb. So that way, again, as I'm not getting enough carbon dioxide out, I'm going to try to then tie up those hydrogen ions in other ways with more buffering, more bicarb, so my pH can stay slightly 7.4 between that range. Okay? All right. Now, if the condition is usually not because of the breath and the breathing rate, but because of things like the kidneys are not working, I am I'm sick and I'm overly diarrhea and I'm losing base fast, or I'm sick and I'm vomiting, or I think I have heartburn and I'm over abusing the what do you call those, Tums, all right? Those can start to lead to conditions where the acid I'm producing, and again, in many cases, it's those fixed and those organic acids, I am producing them at a higher rate than I'm able to get rid of them, and the free hydrogen population is going up faster or down faster than I would like. And so my pH is changing because, again, the system is not working in the ideal rate of production matching excretion, right? And so again, in situations where I am getting rid of the free hydrogens faster than I make them, or I'm retaining and keeping too much base versus the acid I'm creating, I can end up in a situation of metabolic alkalosis. Okay, so again, remember I said if someone overdoes the Tums, so normally in your stomach you have hydrogens, and if every time you put hydrogen you add baking soda and it cancels them out, well, then your cells are like, well, parietal cells are like, I got to put more acid in there. I got to put more acid in there. And again, the minute you put more acid in your stomach, you cancel it out. I got to put more acid in there. I got to put more acid in there. That is how, again, every time I put the hydrogen in, what do I keep? I keep the bicarb. Well, now I'm beginning to, HCO3, I'm beginning to, again, by making hydrogens go into my stomach as fast as I can, I am creating in my system a huge bicarb load that is going to slowly cause me to become metabolic alkalosis, right? So abusing the Tums can do this. Vomiting. So instead of the Tums making the hydrogen ions leave as fast as you make them, if you're vomiting and you're making the hydrogen leave basically as fast as you can put solution in your stomach, that again is going to mean as the hydrogens are pumped in, more bicarb is retained, and I can get myself in a state of metabolic alkalosis. All right, so those are just some of the examples of, again, ways this happens. If the kidneys begin to, again, not able to maybe retain any hydrogens, and I start to have, again, high bicarb amounts, and I'm not able to dump them, the kidneys can be a source of high metabolic alkalosis, right? And lots of drugs, lots of types of different medications that, again, can alter the acid-base balance, and the cells making and using hydrogens and bicarb can also affect this, right? On the flip side, metabolic acidosis would be, again, since I'm usually an acid-making factory, if my kidney nephrons stop working, all right, I can't get rid of these hydrogen ions, okay? And so normally some type of kidney disease is going to cause me to be acidosis. And so this is why a lot of people have to be on, what do you call it, the thing where the kidney, they hook you up to the machine. Oh, I'm blanking. Dialysis. All right, they have to be on dialysis almost every day because the kidneys are impaired in their ability to get rid of hydrogens, and I make a crap load of acid day in and day out, so I need the machine of dialysis to get rid of the hydrogens every day for me. Okay, again, severe diarrhea. Remember that in my stomach, it's all hydrogen rich, and then the minute I jump into my duodenum, I add lots of base, those bases being bicarb, all right? And anytime I add, again, bicarb to the system, I'm going to keep the hydrogens. So if I am, as soon as food enters my small and large intestines, I'm diarrhea out, I run into an issue with dehydration, because the water's not being retained, but I also run into an issue with that severe diarrhea of, as soon as I put the bicarb in and retain the hydrogens, I have to put more bicarb in and retain more hydrogens. And so that can give me severe acidosis. All right. Shock, again, because chances are I'm altering my breathing rate, I'm altering my kidneys, and I'm creating a bunch of acid with the different metabolic actions going on in my cells. So I can get into acidosis. Diet-induced acidosis. I put myself in ketoacidosis. I put myself in a situation, this is very big with diabetes. If I don't control and my body thinks I don't have any glucose, and it starts to put me in a taking fatty acid and trying to make molecules that are useful, that can be maybe utilized in temporary restraint for not having glucose, I'm going to create lots of acids, and those acids can make me ketoacidotic, right? And this can kill me. This is why the keto acid diet or keto diets is a slippery slope to being very dangerous, because if you put yourself in a little bit of ketoacidosis, that's probably not going to kill you, but you are going to be slightly acidic. You put yourself in major ketoacidosis, which could be skip a few extra meals, do a little bit of too much intermittent fasting, and too much maybe dieting extremely. And again, for anorexia, this is an issue, as well as type 1 diabetes and type 2 diabetes. I can get to a place where I become so acidotic that it could kill me. Okay, so these are, again, ways that I can become metabolic acidosis. And again, remember, my runner, my exerciser, if I catch them at the end of the marathon, at the end of the Ironman, they are probably slightly acidotic. Right? The difference is the runner has functioning kidneys, has functioning lungs, and is going to take in oxygen and recuperate and reconstitute those acids in that given second back, hopefully, into molecules that are going to make the acidosis go away. If the acidosis doesn't go away and we continue to add more acid load from other situations or other reasons, that is when the acidosis can be deadly. Okay? Now, one of the essay questions that I ask related to this chapter is, I'm going to ask you, I'm going to say, give me one example of an acid-base disturbance and tell me how did it happen. All right, so if you wanted to choose metabolic acidosis, then you could potentially say, I have kidney disease, so I'm not secreting my hydrogen ions fast enough, so the hydrogen ion load in my body increases, causing me to be acidotic. My respiratory system may try to compensate with a slightly higher than normal respiratory rate, but even with the respiratory system trying to help rid me of excess carbon dioxide, the rate of the kidneys not getting rid of the hydrogens is still greater than what I'm trying to compensate with, and I stay in a constant acidotic state. If you wanted to do, again, something like respiratory alkalosis, I get an anxiety attack and I start to hyperventilate. The hyperventilation makes me breathe off the CO2 faster than I can make CO2 and other acids. So I become, all right, alkalotic, and I will stay alkalotic until either the brain makes me pass out and stops me from breathing and I can recuperate some of my carbon dioxide, or until I can try to slow my breathing rate down with some, again, voluntary control over my breath, or I'll stay slightly alkalotic and slightly hyperventilating, and my kidneys, after a few days of this, might try to kick in and try to help dump more bicarb and retain acid better for me. Okay, so normally I ask you to give me the example of one of these 4 conditions and how it works. How did it get you to that situation? I don't normally ask how compensation occurs, but just so you are aware, start trying to add that last detail. That might be how you help explain what's happening a little bit better. Okay, so that is what I have for you today. All right. Hopefully it has helped explain acidosis. Again, where do the hydrogens come from? Where does the bicarb come from? Why I can end up in a situation of acidosis or alkalosis. When you get to the nursing program, they're going to throw at you numbers from a blood draw, and they're going to throw at you numbers of, you just took someone's vitals and this is how many breaths they're taking per minute, and they're going to tell you with data to tell me what's happening. Well, that's fine if you're ready to go there, but what I want you to do is not look at, this is the value of their plasma bicarb, this is the value of their hydrogens, this is what it says, this is how many breaths they're taking. I want you to back up and think, I am an acid producing factory. This is how I handle it while it's in my system to try to keep the acid from making my pH drop. And then this is how I get rid of it. This is how I try to maintain homeostasis. Okay? And then when you get that mental module in your head of where are the hydrogens coming for, where does the bicarb come, how do they stay balanced, then you can add the numbers, and then you can start to do the little flow chart of diagnosing your patient. Remember, we don't diagnose people, so I don't need you to throw me numbers. What I need you to do is understand, where do hydrogens and bicarbs come from? How is it that I'm kicking them out, using them? And then how is it that I'm temporarily dealing with over excess or under excess of them? And then how does the system try to work itself to keep homeostasis? Okay, so that is what I have for today. I'm going to stop the recording.